My name is Anastasija Zaiko. I'm originally from Lithuania, but now I represent New Zealand, a Sequench Limited company. And in GuardIAS, I'm a marine aquatic biologist, ecologist, but about 15 years ago I started to work more and more in the molecular field using eDNA techniques. And in GuardIAS, as part of Sequench, I participate mostly in the work package 1, related to the data integration, also modeling of the risks and pathways, but also participating in the work package 2, specifically in relation to eDNA methods and eDNA methods application to invasive species detection. So eDNA is a really cool tool because it allows you to detect species without physically obtaining a specimen, catching them. And basically it's like a forensic method when you detect traces of organisms in any environmental matrix. It can be water sample, it can be sediments, it can be biofilm, it can be feces. And you can tell who is present in the environment, what is present in the environment, or has been present recently, and also about the structure of the communities, about the health of the environment. So there are multiple different questions that can be addressed with eDNA. In relation to invasive species, it is a really useful tool because it allows you to upscale the monitoring and detection efforts with very little additional cost compared to traditional methods, and it allows you to get much more resolved information about the presence of potentially risky species. It is really important to plan well how do you collect samples and how do you sample for obtaining robust eDNA-based information on species presence. With the aquatic samples specifically, it is a little bit tricky because we are not sampling from a homogeneous soup of eDNA. It is distributed a little bit randomly and patchy, and therefore most of the common best practices for sample collection and sampling design from ecology and biology applies here. You need to think of replication, you need to think of where to sample, how to sample, what is your research question, so your sampling techniques and approaches and design should be fit for purpose. So you have adequate coverage, spatial coverage, temporal coverage, and also replication. And if from my practice, I know that sampling larger water volumes always help to increase detectability of species that are rare or more randomly distributed. And this is something that we also try to address with developing some new tools and techniques to simplify and also optimize eDNA collection from water. So the main advantage is that it's not destructive. You do not need to capture species. You don't need to destroy habitats by physically taking large amount of material from the environment. You also can detect smallest traces of fingerprints of species present from reasonably tiny volumes or tiny amounts of material you collect in the environment. It's quite sensitive, so you can detect technically individual DNA molecules, although it's not always that simple. You need to consider many other factors that are playing the role in the quality of the sample and also throughout the processing steps, the detectability of this signal. So in general, it's quite useful, cost-efficient and scalable technique. But of course, as any method, it has also some limitations. It cannot answer all questions, and you need to apply it properly and interpret data with the good ecological, environmental knowledge not to make mistakes and not to provide wrong conclusions. Yes, to a certain point, because we are dealing with different environmental matrices. So first of all, the way how we sample differs. Also the material that we obtain differs. Also factors that affect the integrity and quality and amount of eDNA we capture also differs between these different ecosystems. For example, if we are sampling from a small pond, we have most likely more homogeneous structure of the environment, while comparing like a big lake or ocean marine environment where we have much more complex structure of the ecosystem itself. And of course, we have stratification, we have currents, we have multiple different factors that play a role also in the distribution of the species, but also the distribution of the eDNA. In the rivers, we have flow and current that we also need to consider how DNA is being dispersed, especially how it is accumulated in certain points, and also if you catch signal here, it might not be a signal from exactly this location, but coming from upstream. So there are multiple different things that need to be taken into account for, first of all, designing your study correctly, but also interpreting the result you get. I would say in this case, the field is probably in its infancy in terms of the uptake of eDNA, because the eDNA technique is pretty novel still. It's just a couple of decades when this term got introduced. And the first few years it was just purely research and development, but now it's getting already into regulatory structures and concepts. And I know there are a few examples when organizations or decision makers or governmental bodies start implement this as additional tool or as complementary bit of information on biodiversity in general, but also potential spread and occurrences of invasive species. Talking about New Zealand and maybe Australia as well, I can say that these techniques are being uptaken quite efficiently by governmental bodies, by local governments and regional governments and also central governments. And they are already being included into the monitoring campaigns and surveillance campaigns. And I know there are at least a couple of examples when eDNA-based detections actually informed on the arrival of a new species. And then it also helped to promptly react to this incursion and also take some measures to mitigate the spread. And generally, I could say that at the regional council level in New Zealand, this method is quite widely implemented, and it's also very widely used in Australia, both in the biosecurity, by invasion-related programs, but also more widely in the ecosystem health assessment and eco-monitoring.